go ahead.
Okay, sure. Yeah.
And my apologies for the small screen. So thank you for the opportunity. The title is a little different than in the program book, but the particulars are the same. It's really what I'm talking about today is a comparison of evolutionary processes at innate immune loci that sense infection versus innate immune genes that clear infection. I first want to acknowledge the trainees of mine who there we go, who did this work. So Rachel Boyd is now a PhD student at Johns Hopkins. Melanie Denome is a PhD student at Brock University, and Leanne Greaves is now a postdoctoral fellow at McMaster. Now, I always like to start my talks by starting with the bad news, and then hopefully we can build from there. So it's no surprise to anyone here that pathogens are everywhere. They're all around us geographically, although they're not the same geographically. Taxonomically, they're incredibly diverse. They're out to get you, and they evolve really fast. And for all these reasons, pathogen-mediated selection, as we've heard, represents a huge evolutionary force on hosts, and particularly on host immune systems. Now, a lot of what we know about molecular evolution at immune genes and some of the most charismatic stories of balancing and diversifying adaptive evolution come from genes of the adaptive immune system. And in particular, as we've heard today, MHC provides some really compelling examples. We know relatively less about the evolution of innate immune defenses. And that's unfortunate because especially for short-lived animals like small songbirds, like I study, and particularly if they're migratory, we think that innate immune defenses are really crucial to survivorship. So we might ask then, what does pathogen-mediated selection look like at the innate immune genes? And there's two main schools of thought about this. One possibility is that due to evolutionary arms races with fast evolving parasites, we see mainly strong signatures of positive selection at innate immune loci, just as we do at genes like MHC. So processes like rare allele advantage, heterozygote advantage, locally adapted alleles, we might expect balancing selection to maintain high variation both within and perhaps among populations. But another possibility is that selection on innate immune genes may be more purifying than that. And the rationale for that is that the innate immune system is very ancient. It's very crucial to fitness. So we might expect that perhaps there's one best, we might expect that selection has had plenty of time to fix the single most efficacious allele. And so then there might be low tolerance for variance and relatively less variation either within and or among populations at innate defenses. So two possible ways of viewing this, and a third possibility, I guess, and that we can call the functional role hypothesis, is that maybe both of those alternatives, it's mainly positive selection or it's mainly purifying selection, maybe both of these are oversimplifications. So the functional role hypothesis posits that maybe selection acts differently on components of immunity depending on what their job is. So for things like toll-like receptors who are involved primarily in sensing infection, we may indeed see positive selection maintaining high variation within populations. But there's other components of innate immunity that are tasked primarily with clearing the infections. And at these, genes like avian beta defensins, which are antimicrobial peptides, we might expect that purifying selection is purifying selection should be more important, and we might see less variation within and among populations. So to test this hypothesis, my trainees sequenced 3 toll-like receptors, as well as 3 avian beta defensins in song sparrows living in 3 geographic sites in Ontario, Canada. So toll-like receptors, as we've heard, are involved in sensing infection, and for these, we amplified most of the extracellular domain, a region that's especially expected to be subject to balancing selection. We also sequenced 3 avian beta defensins, and for these, we were able to amplify most of the mature peptide. Now, just to get you all oriented, we, of course, at least in spirit, we're here in Prague, but my study site is here in the very southern part of Canada. Here we see a song sparrow. This is the most geographically variable bird in North America. It varies not only in morphology and behavior, but also we now know in terms of disease resistance. So at these 3 study sites here, which we use in this study, we already know that there are geographically confined, locally confined lineages, for example, of avian malaria. And birds are more resistant to their sympatric lineage than they are to avian malaria strains from even just a few hundred kilometers away. And so relative to infection clearing loci, we expected loci that are involved in sensing infection to be under relatively strong positive selection. And so we made a series of predictions, which I'll go through now. So first of all, we might expect simply more variation, a higher proportion of variant sites or a higher proportion of SNPs that are non-synonymous at TLR, the infection sensing loci relative to loci that are clearing infection. The short answer is we do not see that at all. There's no significant elevation in either SNPs or non-synonymous SNPs at toll-like receptors than at avian beta defensins. So we thought, well, what about functional variation? Perhaps the toll-like receptors aren't harboring more non-synonymous SNPs, but the variation they do harbor could perhaps be have more profound effects on protein function. Now, we measured this with a program called Provian. The upshot is that we don't see more functional alterations at toll-like receptors than at ABBD. If anything, it seems to be the reverse. We might also expect a deficit of rare alleles representing positive selection as assessed by Tajima's D-test. We don't see that. If anything, the only locus departing significantly from a d-value of 0 was actually a toll-like receptor that departed in the direction opposite what we predicted. We might expect a higher proportion of sites under positive selection and fewer sites under purifying selection in the toll-like receptors. But I would say these are sort of on the fence. We did see evidence of alignment-wide positive selection at some of the toll-like receptors and not at AVBD. We saw some positively selected codons, but they were a very small proportion of codons. And as well, all the instances of purifying selection we saw were found at the toll-like receptors, and that actually is opposite our prediction. So moving on finally to population genetic structure. We actually did find stronger population genetic structure on average at the toll-like receptors relative to the avian beta defensins that were variable. So this was, and this reflects frequencies of haplotypes that were phased. And when we zoom in and look at specific non-synonymous SNPs, we can see that there's some variation among the different among as well as within different types of immune defense. I want to draw your attention to this point here. That's not actually a bug on your screen. It is an outlier value of FST. So we have a very high FST value for the third codon of toll-like receptor 3. And here's what here's what that variation looks like in terms of the geographic structuring. So this variant at toll-like 3, it's a serine to tyrosine minor allele. It's geographically structured. You see low to 0 frequencies of it in some populations and relatively high frequency of the minor allele in this eastern population. Interestingly, this is also one of the sites that Provian, the effect analyzer, predicted to have a very substantive effect on protein function. So we think it's possible then that this could represent a locally protective allele that maybe protects against some sort of pathogen that may vary spatially over this geographic scale. I'd be hesitant to hang my hat on that. We would want to do functional assays or collect survival information, or at least, and certainly compare this pattern to a neutral locus like microsatellite genetic structure, just to rule out alternative explanations like genetic drift. But at least some evidence that population structure is stronger at least some toll-like receptor loci than at infection clearing loci. So in conclusion, the functional role hypothesis, as I said, predicts that we might see different patterns at defenses that are doing fundamentally different jobs. We did not see strong support for the functional role hypothesis when we looked at processes that are operating within a population. So patterns of variation, or how much functional variation seems to be tolerated, patterns of Tigemus D and site-specific measures of positive or purifying selection. But what we did see was when we zoomed out and compared haplotype and SNP allele frequencies between populations, we did in fact see more structure at toll-like receptors than at AVBD. And this is indeed consistent with the functional role hypothesis. And I would suggest that this may reflect local adaptation to a spatially variable pathogen fauna, although certainly would want to see more studies to confirm that. And finally, certainly we see variation within each of the different types of genes. Not all AVBD genes are the same. Not all TLR genes appear to be experiencing the same patterns. So with that, I will thank the funding sources. Huge thank you to all the people that contribute to this, and thank you very much for your attention. I'm happy to take any questions.